Hi, I'm Carmen Savage. I'm doing A2 level biology, and today we're going to be going over the second video in what could come up in the exam. This one is again on communication and homeostasis. This one's more communication. I know they're in the same module, but I decided to do two different videos because there's like a little bit you have to know and then a big whole page that you had to know. So I was like, if I did that, we'd be here for like half an hour for each video and you'd be getting really bored. So I was like, do them in different videos. Just watch them. You can also tell how long it takes me between videos to another one by the amount of time my hair is drying. Because in the first video it's still a lot wetter than this. But because I'm on YouTube at the same time, I get distracted. But I think I've been productive because I've watched like 3 Jenna Marbles videos, 3 Max No Sleeve videos, and I clicked on this one. She was quite funny actually, but I closed her by accident. I don't know who it is. It was like this girl, she was telling me all about her Christmas presents, and there's like a Q&A with her, and she was in a hurricane. It was really cool. And I'll have to go back and find her because I liked her videos. But anyway, back to revision, because I'm getting really distracted. We're going to start with sensory receptors. The things we have to know in communication that haven't come up before are sensory receptors and the resting potential, and like how that stays. So it's going to start off with sensory receptors. Now, what are sensory receptors? Well, they are specialised cells that detect changes in our surroundings. They are energy transducers, and they convert energy from one form to another form. And each type of transducer is adapted to detect changes in a particular form of energy. So this could be a change in, the light levels, a change in pressure on the skin, or another energy change. And each change in energy levels in the environment is called a stimulus. And whatever the stimulus is, the sensory receptors convert the energy into a form of electrical energy called a nerve impulse. So, yeah, great. So that is sensory receptors. I can't work out if I should go over nerve impulses and motor neurons all in the same video, because that is a lot of things to do. No, what I'm going to do is this is what happens when I don't plan what I'm going to say in my videos. I get confused. Okay, what I'm going to do is I'm going to say the receptor, and then you have to try and work out what energy change they detect. And then I'll say it, obviously. And then you'll be like, yeah, I got it right. Or, oh, crap, I got that wrong. Okay, so if your receptor was a light-sensitive cell in the retina of the eye, what would that detect? So, the light-sensitive cell in the retina of the eye, that detects light intensity and range of wavelengths. Okay, next one is the olfactory cells lining the inner surface in the nose, in the nasal cavity. What would that detect? That would detect the presence of volatile chemicals. They have said that in a very confusing way. It's like smell, I don't know. Presence of volatile chemicals, great. Next one, taste buds on the tongue. Hard palate, epiglottis, and the first part of the esophagus. What could that be? What could that be on my tongue? The presence of soluble chemicals. Next receptor, pressure receptors. Pacinian corpuscles in the skin. Touching my skin. What could that be? Obviously, pressure. They detect pressure. The energy change detected is pressure. Woo! Next one, sound receptors in the inner ear. Ooh. The cochlea. Ooh, what could that be? Vibrations in the air. The final one we're going to say is the muscle spindles. And the energy change they detected is the length of muscle fibres. Because, it changes. Yeah, the length of the muscle fibres. Super duper! So that was sensory receptors. Yay! So now we're going to go over the resting potential and the resting neuron. So what is a resting neuron? Well, when a neuron is said not to be transmitting an action potential, it is said to be at rest. But in fact it's actually actively transporting ions across its cell surface membrane. And sodium potassium ions use ATP to pump 3 sodium ions out of the cell for every 2 potassium ions that are pumped in. And the plasma membrane is said to be more permeable to potassium ions than sodium ions, and many diffuse out again. The cell cytoplasm also contains large organic anions. What are anions? Anions are negatively charged ions. Okay. I don't remember getting taught about anions. Probably did. But yeah, so anions are negatively charged ions. Super duper. And that means that the interior of the cell is maintained at a negative potential compared to the outside. And that means that the cell membrane is said to be polarized. And so the potential difference across the cell membrane is about negative 60 MV. And that is the resting potential. So if we want a definition, key definition of the resting potential, say that the resting potential is the potential difference or voltage across the neuron cell membrane while the neuron is at rest. It's about negative 60 MV inside the cell compared with the outside. Other cells might also maintain a resting potential that might change under certain circumstances. So maintaining resting potential, pumping 3 sodium ions out of the cell for every 2 potassium ions that are pumped in. Yay! Woo! So that is resting potential and sensory receptors. Wasn't that long of a video. If you want to, if you've heard that, what I've just said, and then you're like, oh, but so that's the resting potential. So how does an action potential occur? Or what's an all or nothing? What does that mean? What's the all or nothing law? Because you remembered it from that. Then go and watch my other videos, because I've done videos on them. If you want to, I was talking about a minute ago if I was going to talk about generating nerve impulses or generated potentials, but I didn't. So if you want to know about them, then just go back to my other videos, because I've talked about them there. Yeah. So good luck in your exam. If you're watching this in 2013 and you got it tomorrow, like me, then super duper good luck. Remember to revise everything. Don't just revise these videos, because loads of stuff will come up. Yeah, so good luck, and I hope you do really well. Bye.